Hey there, guys. Mr. Gonzalez here with another review for our test. This is a quick review of the cell. Just follow along in your notes. Pause the video if you need. Repeat 5,000 times. So here we go. This is what we talked about when we discussed the cell. We started by going over the parts of the cell. This diagram, which I painted myself, shows all the parts, but ours on the board sort of looked a little better. This is what it looked like. So in your notes you have this, and what I may ask you to do on the test is I'll have a cell that's pretty much either blank or has some of the organelles, and you'll have to fill them out. You may see this picture and you'll need to label some of the organelles, so be either ready to draw some of it, label some of it, and of course say what the functions are, which we'll go over now. Now, a plant cell is totally the same as the animal cell we drew. Couple of differences: they don't have flagellum or cilia. They don't have centrioles either. Something else you'll see is the plant cells have a chloroplast. They have a cell wall. Now remember, they have a cell wall and a cell membrane, not just a cell wall. And they have a very large vacuole which holds food or water or anything like that. Okay, so let's take a tour of the cell. Definition for the cell was the basic unit of life. That basically means that it can either build living things, living organisms, like it's the smallest brick that can build up an organism, or it can actually be, that's a subunit of tissue, by the way, if it builds up organs. But it also can be the smallest thing that's alive. You can have single-celled organisms like bacteria or protists. What are the two types of cells? There are two types of cells, prokaryotic and eukaryotic. That's what how you describe them. They're also known as prokaryotes and eukaryotes, if you see that as well. Main difference, eukaryotes have membrane-enclosed organelles, so they have a nucleus, they have mitochondria as a membrane-enclosed, vacuoles are membrane-enclosed organelles, and prokaryotic are basically just a bunch of jelly. No, they have a cell wall, cell membrane, and they have a few organelles like ribosomes, but the main thing is they don't have a membrane-enclosed nucleus. They're also a lot smaller, organelles. So here we go. Functions of the organelles. The nucleus, which we consider the brain of the cell, controls all cell activities, also holds the genetic material of the cell, so the DNA is inside the nucleus. Nucleolus, that's that purple dot you see in there, that's inside the nucleus, and its job is to make ribosomes. Ribosomes are drawn as like little dots, either inside the cell or like in the cytoplasm or on the endoplasmic reticulum. So they're usually put on the endoplasmic reticulum, the ER, because they help, they make proteins. That's their function. And the ER usually packages and processes those proteins. Mitochondria, we've been talking about recently. It's the powerhouse of the cell. It's where ATP is made. It's where glucose is processed to make ATP, energy for the cell. The ER, like I said, packages and processes proteins. It's also one big crazy membrane. And the Golgi apparatus transports a lot of these proteins that are made by the ER. So the ER makes like these little membrane vesicles. If you look in the lower right, it shows like proteins being packaged in this little transport vesicle. A vesicle is like a vacuole. And then the Golgi apparatus grabs that vesicle and sort of transports it out by exocytosis. Thank you. Lysosomes are membrane-enclosed vesicles that have little enzymes in there which help the cell digest stuff, and vacuoles are for storage. Cytoskeleton gives the cell structure, so the cell would collapse on itself if it didn't have all these this cytoskeleton. Centrioles are the little dudes that make the cytoskeleton. They're also key in mitosis because they form the spindle fiber, which are what we talked about, the cables that sort of make the chromosomes separate during cell division. Cilia and flagellum help in movement. And then we talked about the cell membrane. there were a couple of things we talked about the cell membrane. The first thing is we talked about its structure. We said, hey, what is some stuff that can go in and out of cells? We said water, CO2, O2, waste nutrients, and then we basically said the cell membrane is like a bouncer at a club. It decides who comes in and out of the club. It's selectively permeable, or has selective permeability. This means that it decides who can enter and who can exit the cell. So, we said there's a couple of types of movement that occur. The first one is endo or exocytosis. This is if the particle's really, really, really big, like the dark green one on the right. The membrane circles and encloses a large food particle, makes a vesicle, and usually there's some kind of fusion that occurs with some other thing, like a lysosome. And the lysosome has enzymes in there that help break up, digestion occurs, and then exocytosis of waste can occur. So this picture right here shows you both. Be prepared to draw endo or exocytosis. Next, we talked about what the membrane sort of looks like. Should have did this first, sorry. What the membrane sort of looks like. We use the basketball as an analogy because basketball has all these little dimples in there, and so does a cell. The cell has all these little dimples called phospholipids. They're all these like little balls that can float around. They also have these big purple beans in there. Those are transport proteins which help larger particles move through that are usually pumped in and out of the cell. Some things can go through the little balls, like through the phospholipid bilayer. Other things have to go through the protein. And some things are too big to go through either. The second type of movement is diffusion. Now, diffusion occurs naturally. Diffusion is like the spreading of molecules. Like if you're at a department store and they have like the perfume and the lady comes up and she's like, you want to try this? And she sprays it on your wrist and you didn't really want it. It diffuses out so everyone smells the odor. That's diffusion. Now, if you put a membrane in the way and the membrane is semi-permeable, that means stuff can go in and out. If you spray or if molecules move, they'll go through the membrane. And they'll move until everything's equal on either side, and then diffusion stops. Okay? Now, what exists between the membrane is called a concentration gradient. Concentration gradient is like there's the amount of particles differs from the inside or outside. So there's a difference in concentration. So there's a gradient that exists. Now, in diffusion, the movement of the particles goes down the gradient, which is like downhill, kind of. Stuff that moves against the concentration gradient is active transport. Okay, so that's the other movement. So in this picture, does the molecules move to A to B or B to A? A to B or B to A? A to B. Yeah. Oh yeah. Okay, so we talked about where does diffusion happen in us. We said in our lungs is a perfect example because you breathe in oxygen. All that oxygen goes to the end of your lungs called the alveoli, which are tiny sacs that are surrounded by blood vessels. The oxygen totally diffuses in. Why? Because your blood has no oxygen at that point. So all the oxygen that's at your lungs, there's a high concentration of that oxygen. And so it automatically goes from high to low, low being your blood. Now CO2 is the opposite. Because you do cellular respiration, which makes CO2 as waste, it builds up in your blood, high concentration in your blood goes to the alveoli, and then it diffuses out automatically. And so diffusion occurs at the end of your lungs. Active transport, that's the third. Now this goes against the gradient. So instead of high to low, this goes low to high. Now the only difference, or one big difference, besides low to high, is that it needs energy to occur. Okay? So think of this awesome slide at a playground. Okay, so one good analogy is that diffusion is moving down the slide, down the concentration. You don't need any energy, and so you could just slide down. But active transport is if you had to go against the gradient and do work. You need to expend energy to go up the elephant's butt. Okay, so where does active transport occur in us? Well, we said, hey, the small intestine is a great example. Glucose has to get in all your cells, and so it goes down your small intestine into these little things, these little hairs called villi. And so in this example, we have a bunch of there's the phospholipid bilayer at the bottom. The little white hexagons are sugar. So we start off intaking glucose as diffusion. Right? Then diffusion stops. Why did diffusion stop? Well, because it's equal on either side. We said diffusion stops when there's the equal concentration on either side. So then active transport takes over. You need to start pulling those glucose molecules in, and now it's going low to high, and so that takes energy. Osmosis! Now, the ones we've been talking about have been movement of particles, of things, molecules. This one is also the movement of molecules, but water, some sort of solution. So look at this. If you had dirty water on one side and clean water on the other side, the dirty water would actually have more. If you put a semi-permeable membrane between them, the dirty water would have a higher level. Now think about that. Why? Why would the clean water be lower? Well, water always moves to where there's more stuff or more particles. So that clean water totally moved to the left, to the red side. Water will always move to where there's greater concentration of molecules. Cool. So osmosis. Oh, I just did this. Okay. So here are some cells. You take some red blood cells, you put them in 3 solutions. The one all the way on the left is called an isotonic solution, a solution that's totally the same amount of salt inside and outside. The second one is a hypertonic solution. That's one that's so salty and the inside is not salty inside of the cell. And the third one is a hypotonic solution. That's a solution that's less salty or has a less concentration of something than the inside of the cell. So you have to know what happens in each one. In the first one, if I put a cell in the same sort of concentration solution, nothing happens because it's equal in and out. In the middle one, if I put it in a solution that's extra salty, well, stuff, water, always moves to where there's more salt or more ions or more stuff. So that cell shrinks. And the one all the way on the right, well, the fresh water, since the cell is saltier inside, the water always moves to where there's more concentration of something, so the water is going to fill up those cells and they blow up. So, we said this question. Hey, if you had fish, they have to sort of control their body water all the time, so one of them drinks all the time and one of them pees all the time. Okay. So, who drinks and who pees? So, the saltwater fish on the left, also known as Nemo, his surroundings are saltier. So is he losing water or is he gaining water all the time? Well, since he's fresher than the water around him, he's losing water all the time, so he needs to drink. And the freshwater fish is the opposite. The surrounding water is fresher or less salty than his body, which has a higher concentration of stuff, and so water automatically goes through his body and starts filling up his cells, so he has to pee all the time. And that's it for the cell. Good luck, guys.